Back friends, welcome back in another video tutorial for microbiology with Chaknik. So in this video tutorial we are going to discuss a previous year CSIR net question from recombinant DNA technology, that is based on our first chapter recombinant DNA technology, and it is mainly a question from the screening procedure. I have discussed with you about the screening procedure multiple times. So today we are going to see a question from that screening problem. So first of all, let's read the question. So they tell us that in order to clone an eukaryotic gene in PBR322 plasmid vector, that means PBR322 plasmid vector, and in that PBR322 plasmid vector we are going to insert a gene, right? The desired DNA fragment was produced by PST1 cleavage and incubated with PST1 digested PBR322. That means what? That means that same restriction endonuclease here used to cut the gene of interest along with to cut the plasmid. Now PST1 cleavage site lies within the ampicillin resistant gene. What does that mean? PST1 cleavage site lies within the ampicillin resistant gene. That means here the ampicillin is the secondary marker, right? Secondary selection marker, and it is of negative selection or insertional inactivation, okay, and ligated. So mixture of the ligated cell were used to transform E. coli and plasmid containing bacteria were selected by their growth in tetracycline containing medium. You have to focus on this. So they are telling us which type of plasmid or plasmids will be found. So let's see. So they tell us that they have used PBR322 as a vector. Okay, very good. Then a gene of interest, okay, and it is basically a eukaryotic gene of interest, and both eukaryotic gene of interest and the PBR32 is cut with PST1. Okay, very good. Now in presence of ligase, they forms the recombinant DNA, but here three possible options can be possible. What possible options? Let me discuss with you. So first of all, the plasmid can attached with the gene of interest. That is our required option. Okay, very good. Plasmid can self-ligate in presence of ligase. That can also be possible. And another important thing, as here we are not inserting only the one plasmid, right? There are multiple plasmid present. That means here in a solution we are putting plasmid multiple types of along multiple in multiple numbers and along with multiple number of gene of interest, right? So there can be a possibility that two plasmid can attached with together to form a big plasmid. And this joining of two plasmid is called, according to this question, is concatenation. Okay. Although concatemer is the term of hydrology, that is the DNA molecule packaged during the fudge head mechanism, I know, but here they are using the term concatemerization to simply tell that two plasmid here attached with each other to form a big plasmid. Okay, so these three type of possibilities can happen, as here we are using tetracycline, right? So as our second antibiotic resistant gene is ampicillin, so if we plate them in ampicillin, then only the non-recombinant colonies will grow, but here all type of possibilities can possible. That means gene of our plasmid vector along with the gene of interest is the one possibility. Recircularized plasmid is another possibility, and the big plasmid can occur, which is the result of concatenation, and that is why the answer will be C, that is circular PBR322 plasmid containing the target gene and resistant to only tetracycline, because it is sensitive to ampicillin, as PHT cleavage site lies within the ampicillin. And a recircularized plasmid can form which is resistant to both ampicillin and tetracycline, and concatenarized PBR322 resistant to both ampicillin and tetracycline can also form. So this kind of three possibility can occur in such cases. If they tell you to tell which kind of plasmid can you observe, which you are going to observe in case of both ampicillin and tetracycline containing medium, so in that case your answer will be concatenarized plasmid containing both ampicillin and tetracycline resistance, and recircularized plasmid containing both ampicillin and tetracycline resistance. Here the recombinant DNA cannot be obtained, as here tetracycline antibiotic is also ampicillin, and antibiotic is also present along with the tetracycline, and as ampicillin resistance property is inhibited upon insertion of gene of interest, so our recombinant DNA containing colonies cannot grow. That is the idea to answer this question. So this is the CSI and NET question. So I hope that you have learned it well and you have understood it well. So thank you.